Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 26 from our Brock Biology of Microorganisms textbook. This chapter deals with innate immunity, also known as non-specific immune response. Now, what you should know is immunity is the ability of an organism to resist infection. And there are two branches to immunity. There is what's covered in this chapter called the innate immune system or non-specific immune system. And then there's the stuff from the next chapter, which we will cover in the next video, chapter 27, and that covers the adaptive or specific immune system. These are two branches, and what I want you to know is this about innate immunity. Chapter 26 covers innate or non-specific immunity. This is a type of immunity that does not require previous exposure to a pathogen or its products. It does not get better over time. It does not remember previous insults. The non-inducible ability to recognize and destroy an individual pathogen or its products. So this innate immune system is just as good today as it was when you were born. It doesn't improve over time. It doesn't remember previous insults or previous pathogen exposure. It just remembers basically nothing. It's a baseline resistance. You can think of it that way. And so anything that helps your body to prevent infection, anything that prevents you from getting an infection that doesn't get better is covered by your innate immune system. So we're going to talk about that. And in chapter 27, we're going to discuss adaptive immunity. Remember, this is also known as specific immunity. This is the acquired ability to recognize and destroy a particular pathogen or its products. This is the reason why you get a vaccine. This is the reason why you can't get sick with the same pathogen twice. Your immune system remembers previous insults, remembers previous antigen. What is an antigen? So adaptive immunity is directed towards an individual molecular component of a pathogen. So a molecular component of a pathogen. So for example, a spike protein from a virus or a surface protein from a bacteria, if these are introduced in the form of a vaccine, then the immune system will remember that protein, that molecular component, that antigen for next time. It is dependent on previous exposure to pathogen or its products. So adaptive immunity improves over time with each subsequent infection. So again, there are two branches to immunity. Chapter 26, we're dealing with innate immunity. These are the phagocytes, which are the primary effector cells. Dendritic cells, neutrophils, and macrophages are the phagocytes. These are part of the innate immune system, and there's much more to the innate immune system than these three phagocytes, but we're going to talk about these three phagocytes. You should know that innate immunity is rapid response within several hours, and they respond to general broad-range pathogens. There's no immunity here after exposure. Conversely, chapter 27, we're dealing with adaptive immunity. This is immunity that gets better over time. We're dealing with what are called lymphocyte white blood cells, lymphocytes, which are primary effector cells here. These include the B cells and the T cell lymphocytes of the immune system. These pose a focused attack on a specific pathogen. The B cells form plasma cells which secrete antibodies against the pathogen or against the antigen, and the T cells can also recognize the antigen and attack. And then this is the memory part of the adaptive immune system. Post-exposure immunity by B and T memory cells is common. These B cells and T cells can differentiate into memory cells whose job it is to linger in the body and remember that exposure for next time. And this is a type of response that requires several days. This is why when you get your vaccination, for instance, you have to wait several days for the full effect to occur, because the B cells and the T cells have to recognize that antigen, they have to mount a host immune response, produce the antibodies required to fight the infection. So what are some barriers to pathogen invasion? You've got natural host resistance. So for example, even the normal microbiota in your gut, in your mouth, on your skin, normal microbiota is part of your innate immune system. It helps the host resist pathogens. How? Mainly by taking up space, right? Particularly on the skin and the gut, by competitive exclusion. You've got friendly, probiotics in your gut and on your skin, and these bacteria help exclude more harmful bacteria. Remember, we discussed earlier that if you take too many antibiotics, you could clear out some of your gut flora, and that could free up real estate that other more harmful bacteria like Clostridium difficile can come and take up residency and damage the gut lining. The ability of microbes to cause disease varies between species. Remember, there are some microbes that are more virulent and more pathogenic than others. So here you can see these are all part of the innate immune system. Remember, I said anything that doesn't get better over time but helps to keep your body free of infection is part of the innate immune system. So that includes skin. Your skin is actually part of the innate immune system. Why? Because it's a physical barrier and it produces antimicrobial fatty acids, antibacterial peptides. There's a normal microbiota. So your skin, think about it, your skin is the first line of defense. Things need to be able to penetrate through the skin to cause an infection. So your skin does a great job at preventing infection. Also, you could have removal of particles by the cilia and the nasopharynx. You've got stomach acidity. That's part of your innate immune system. It inhibits microbial growth. So if you were to ingest some harmful bacterium, the stomach acids can kill it. There's, again, normal microbiota as part of your innate immune system, because the normal microbiota can outcompete the bad microbiota. Flushing of the urinary tract prevents infection like UTI infections. Lysozyme is an enzyme in the tears and other secretions which dissolves bacterial cell walls. It's an enzyme that's secreted by your glands in order to destroy cell walls of bacteria. Mucus and cilia lining of the trachea. Remember, I've said earlier that if you inhale dust or inhale bacterium, you have the mucociliary blanket in your guts. In your trachea and your lung lining that can push that debris out of the lungs, flush it down to the stomach where the acids can destroy the bacteria. Mucus antibacterial peptides. So you've got mucus. Mucus is really good at preventing infection because it not only flushes, but it has antibacterial peptides in it. And phagocytes. Phagocytes are cells that can endocytose bacterium and other pathogens in lungs to prevent infection. In fact, in your lungs, in the alveoli of your lungs, you have these specialized macrophages which can phagocytose any invaders in the distal portions of the lung. You have blood and lymph proteins, including the complement proteins and the white blood cells that can inhibit microbial growth. You've got a rapid pH change, from the acidity of the stomach to the more neutral conditions of the small intestine. You've got a rapid pH change which inhibits microbial growth. Again, your epithelial cells of your skin are constantly sloughing off, shedding off, which helps prevent bacteria from sticking onto you. There's also tight junction between the skin cells, which makes it watertight and also impervious to bacterium unless they have some kind of enzyme that can chew up the skin, like hyaluronidase, etc. So again, remember infection site and tissue specificity. If a bacteria or virus or pathogen cannot stick to you, then it will not infect you. Different pathogens invade different tissues. The routes of infection are crucial. So again, remember, a virus or a bacteria needs to be able to adhere to your cells, colonize, grow, invade, invasiveness, etc. Remember, there were steps to infection. Again, speaking to physical and chemical barriers to infection, mucosal membranes are coated with a thick layer of mucus. The mucus helps to prevent infection. Stomach acid inhibits bacterial growth. Skin is salty and acidic, limiting bacterial growth. It also sloughs off, right? So it's very difficult to grow on the skin. Okay, let's talk about some of the cells and the organs now of the immune system. So here we're talking about the blood and the lymphatic systems. The lymphatic system is a separate circulatory system that drains lymph fluid from extravascular tissues. Blood is pumped through the arteries and capillaries and returns to the body through the veins. And then you've got these capillary beds where leukocytes, and by the way, leukocytes mean white blood cells. In capillary beds, leukocytes and solutes pass from the blood into the lymphatic system. So you've got the lymphatic system, which is its own circulatory system of white blood cells. And then you have your regular circulatory system, which is, you've got blood pumping around, you've got blood proteins, serum, plasma, you've got white blood cells in there, and they can kind of speak to one another in that they can exchange white blood cells between the two systems. Lymph nodes contain high concentrations of lymphocytes, and phagocytes. So let me show you something. Here is an image of your lymphatics. You've got the thymus, which is a lymph tissue, or I should say an immune organ. The thymus, a butterfly-shaped organ here. You've got the bone marrow. Remember, the bone marrow is the site of hematopoiesis, where hematopoietic stem cells give rise to the white blood cells and the red blood cells. You've got the spleen, which is a major organ in the immune system. You've got the lymph nodes all over the place. You've got mucosa-associated lymphoid tissues, which are called MALT. You've got the thoracic duct, the lymph nodes, and look how the lymph nodes are all connected to one another. You see this green, all these green lines everywhere? These are your lymph circulatory system. The lymph nodes are connected via ducts to one another. And here you can see that there's these lymph capillaries that lead to lymph vessels, to the lymph nodes, to the circulatory system. Then you have the circulatory system, which goes from the heart to the lungs, and then back to the, through the veins, back to the heart. And there are locations in the body where the blood capillaries meet with the lymph capillaries, and there's an exchange of white blood cells. See here, you've got the capillaries, the blood capillaries on the left, you've got the lymphatic capillaries on the right, and you can have an exchange of white blood cells through diapedesis, which is the process by which white blood cells like neutrophils and macrophages and such can squeeze through the endothelial, the blood vessels. These white blood cells can actually move from capillary to lymphatics and vice versa. Okay, so I keep saying leukocytes. Remember what leukocyte means is white blood cells. White blood cells. And about 0.1% of your blood cells are the white blood cells. These include the phagocytes and the lymphocytes. Phagocytes, remember, were your macrophages, neutrophils, and dendritic cells. Lymphocytes were your B cells and your T cells. and also your natural killer cells. We're going to talk about all these different types of cells in a little bit. So whole blood is composed of plasma cells and plasma and cells. Plasma contains proteins and other solutes. Serum, on the other hand, serum is the portion of blood that is not cells or clotting proteins. So just a little bit of definition for you. Again, here are the major types of cells found in human blood. You've got erythrocytes. These are your red blood cells. And you see they make up a large majority of blood are the red blood cells, the erythrocytes. Then you've got the leukocytes. Remember, this means white blood cells. And this makes up a minority of the cells in your body, of your blood. The lymphocytes, remember, these are the B cells, the T cells, and the natural killer cells. The granulocytes and the monocytes. We're going to talk about these as well. So again, what are leukocytes? Leukocytes are nucleated white blood cells. These are all of your white blood cells, which includes the lymphocytes, which are specialized leukocytes involved exclusively in adaptive immune response. Remember, these are your B cells and your T cells, specifically the B cells and the T cells. We're going to be going through a lot of detail about the B cells and the T cells in chapter 27, because these are the two big players in the adaptive immune response. We're going to talk about that. B cells, these are lymphocytes that originate and mature in the bone marrow. T cells, I want you to know that they originate in the bone marrow. However, they mature in the thymus. Remember, the butterfly-shaped organ near the throat is the thymus. So the bone marrow, because B cells and T cells mature in the bone marrow and the thymus, respectively, the bone marrow and the thymus are primary lymphoid organs. They're considered primary lymphoid organs. So you can see here the red bone marrow. You've got the bone marrow, and the bone marrow is the site of what's called hematopoiesis. That's where blood cells come from. They're born from these cells called hematopoietic stem cells. These are stem cells that give rise to all the different types of blood, including the red blood cells, the platelets, and all these different white blood cells, okay? Hematopoietic stem cells give rise to all the different types of blood. These are all of the white blood cells. These are called the white blood cells, or leukocytes. And here's how it works, okay? The hematopoietic stem cells give rise to two types of progenitor cells, the myeloid precursors and the lymphoid precursors. The myeloid precursors can give rise to the dendritic cells, the macrophages, the neutrophils, and the mast cells. And on the other hand, the hematopoietic cells could give rise to lymphoid precursors that can give rise to natural killer cells, T cells, and B cells, which can differentiate into plasma cells. So looking at the white blood cells at the bottom, the dendritic cells and the macrophages are called antigen-presenting cells. They're antigen-presenting cells. And by the way, the B cells are also antigen-presenting cells. The dendritic cells, macrophages, and neutrophils are called the phagocytes. These are the cells that can phagocytose or endocytose foreign invaders. Then you've got your mast cells. By the way, the neutrophils and the mast cells are called granulocytes because they have granules inside, which they can release, and these granules can contain all kinds of things like hormones. They can contain histamine. They can contain perforins and other types of granzymes, things that can damage cells. Basically, depending on the white blood cell, you have a different type of granule inside that you could release. And by the way, not listed here, but also important to know about, are the eosinophils, neutrophils, and basophils. Oh, here's neutrophils, but eosinophils and basophils are examples of granulocytes as well, in case you're wondering. This is not a comprehensive list, by the way, of all of the white blood cells in your body. There's more. There's, you know, the basophils, eosinophils. What's the other blood cells here are not, the red blood cells are not listed as well. You know, the erythrocytes and also the platelets are not listed. So this is not an exhaustive list here of the white blood cells in your body, just so you know. And again, let's look at this branch here, the lymphoid precursors. These give rise to the B cells and the T cells, okay? There are T cells. There's two forms of T cells. There's helper T cells and cytotoxic T cells. We'll talk about those in chapter 27. There's natural killer cells, which are part of the innate immune system. And then there's the B cells, which become the plasma cells. So lots and lots to think about here. And by the way, remember I said the T cells and the B cells? These are the cells of the adaptive immune system, which means all the rest of these are part of the innate immune system. All right, I know that was a mouthful, but let's keep going. The myeloid cells, remember the myeloid precursors. The myeloid cells are these ones here in red. The myeloid cells are derived from the myeloid precursor cell, can be divided into two categories, either antigen-presenting cells, which what these cells do is engulf, process, and present antibodies to lymphocytes. These include the monocytes, the macrophages, and the dendritic cells. And remember, antigen-presenting cells do also include the B cells. Granulocytes, so we just talked about these guys here, the dendritic cells, macrophages, and monocytes. Monocytes become macrophages, by the way, after a little bit of circulating in the blood. The granulocytes contain toxins or enzymes that are released to kill target cells. Remember, the granulocytes can release granzymes, can release those granules to have effects on the body. You've got the neutrophils, basophils, eosinophils, and also the mast cells are a type of granulocyte, but they don't release their granules to damage other types of cells. They release their granzymes, which are full of, they release their granules, I should say, which are full of histamine to help, you know, with inflammation, to help open up the blood vessels, cause vasodilation, open up the pores of the blood vessels to allow neutrophils to migrate in. Then the lymphoid stem cells, remember the lymphoid stem cells, the lymphoid branch here, these include the T cells and the B cells of the adaptive immune system, as well as the natural killer cells of the innate immune system. Each lymphocyte produces a unique protein that interacts with a single antigen. The T cells, they present a unique protein on their surface called the T cell receptor, or TCR. The B cells, they have a unique protein on their surface too, which is called an antibody or immunoglobulin. The shorthand for immunoglobulin is Ig. All right, moving on to phagocytic response. How does phagocytosis work? So microbial invasion, and by the way, when I talk about phagocytosis, I'm talking about these phagocytic cells. I'm talking about the dendritic cells, macrophages, and neutrophils, and to some extent the monocytes. That's what we're talking about here. Microbial invasion is the ability of a pathogen to enter the host cells or tissues, multiply, spread, and cause disease. Remember, we covered this in the previous chapter. Invasion is the ability of a pathogen to enter host cells or tissues, multiply, spread, and cause disease. Tissue damage triggers the recruitment of a large number of phagocytes. Tissue damage and cytokine release. Cytokines are signaling molecules. They're little signaling molecules that are secreted by one type of cell and which have receptors on other cells. Tissue damage and cytokine release. Resident leukocytes, so these are white blood cells that live in particular tissues, Resident leukocytes and damaged cells release cytokines. These are those like signaling molecules or chemical mediators that allow communication between white blood cells. That's how white blood cells communicate with one another. One white blood cell type will release a cytokine. That cytokine has a receptor on a different white blood cell type, and that can recruit white blood cells to the area. That could do a number of things. Release of cytokines and chemokines allows the drawing in of macrophages and neutrophils. Remember, these are the highly phagocytic cells of the immune system. Macrophages and neutrophils follow the chemokine attractant to the area as they leave circulation. The term for leaving circulation, when the macrophages and neutrophils squeeze through blood vessels to get to a particular location where there's an infection, that's called extravasation, also known as diapedesis. So here you can see there's a wound, like a blade cut through the skin. This introduced some bacteria to the location. There's a chemokine and chemokine gradient formed. So these damaged tissues release chemokines, and that can cause acute inflammation. That will recruit neutrophils, macrophages, other white blood cells to the area so that they could phagocytose the bacteria and prevent infection. Here you can see the process of extravasation, also known as diapedesis. Look, there's bacteria in the area. Those chemokines have released, drawing the white blood cells to the area. Notice the white blood cells normally circulate in the blood or lymphatics, but upon infection and acute inflammatory response, the white blood cells can actually squeeze themselves through the endothelial vessels, through the blood vessels. Look, this is called extravasation or diapedesis. Though this neutrophil is squeezing through the capillary, and then once here in the interstitial fluid where the bacterium are, it can phagocytose. It can gobble up these bacteria. It can phagocytose. So neutrophils can do this, macrophages can do this, and the red blood cells just keep on doing what they do, circulating in the blood. So how do these phagocytes, how do they know what to gobble up, right? What the bacteria are, so they can endocytose them? Well, it turns out that there are receptors on the surface of the phagocytes called pattern recognition receptors. So these are receptors on the surface of the neutrophils, the macrophages, the neutrophils, macrophages, and dendritic cells. They have receptors on the surface, and these receptors can respond to patterns that they recognize. These patterns are called PAMPs, or pathogen-associated molecular patterns. Again, did you see what I said there? These patterns, called pathogen-associated molecular patterns, are recognized by receptors called pattern recognition receptors on the surface of the phagocytes. What are these PAMPs? Pathogens have structures and molecules not found in or on the host cell. For example, peptidoglycan, flagellin, double-strand RNA. So take a look here. Here's some pathogens. Here's some pathogens. It looks like 3 rod-shaped bacterium and a spiral-shaped bacteria. This is a phagocyte, and notice how it has these PRRs. Remember, pathogen recognition receptors that recognize patterns on the bacterium. And then phagocytosis can ensue. Again, what types of things does the PAMP recognize? Things like peptidoglycan, flagellin, double-strand RNA. These are things that should not exist in your body. So once phagocytosis happens, we can form a phagolysosome. So when engulfed, a bacteria will be held in a membrane, right? Because when you phagocytose something, you bring that something in as with part of your membrane around it. This bound vesicle is called a phagosome. What you can then do is fuse the phagosome with a lysosome. Remember, the lysosome is a digestive organelle in the cell. It's full of hydrolytic enzymes. Hydrolytic enzymes can chew up biomolecules. So what you would do is fuse a lysosome with a phagosome, forming a phagolysosome, and now you can digest the phagocytosed bacteria. Phagocytes also produce toxic reactive oxygen intermediates to kill the bacteria within a phagolysosome. Remember reactive oxygen intermediates? We talked about those. Those are called ROS, R-O-S, reactive oxygen species. These include like hydrogen peroxide, singlet oxygen, etc. So the phagocytes can also produce ROS to help damage and destroy the phagocytosed pathogen. All right, so before we carry on with any more of this, let me take a quick break to rest my voice. We'll take a quick little break time with Gizmo and Wicket, see what they're up to, and we'll be right back with more. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's keep going with chapter 26. Remember, we were discussing the importance of phagocytosis in the immune system with the phagocytic cells. However, there are mechanisms by which microorganisms can inhibit phagocytes. So here are some examples. Mycobacteria tuberculosis, the causative agent of tuberculosis, produces carotenoids, and these help to neutralize the singlet oxygen in the phagosome. And it has, remember, Mycobacterium have the waxy cell wall. That's what makes them acid fast, those mycolic acids. This waxy cell wall absorbs free radicals. This pathogen, therefore, can live and divide within the phagocytes. So this is why it forms such a persistent infection inside of your lungs. The Mycobacteria tuberculosis gets into the lung. Remember, I said there are macrophages at the alveoli of the lung that can gobble up invaders. Well, the tuberculosis gets phagocytosed. However, this creature can live inside of those macrophages, and that's how it becomes a persistent lung infection. The pathogens such as Streptococcus pyogenes produce leukocidins. Leuko means white blood cells. Cidins means killers. So these are leukocidins, which are these molecules that can kill the white blood cells. Dead white blood cells are found in pus. So when you kill the white blood cells, you kill the phagocytes, well then the phagocytes can't gobble you up. Lastly, some pathogens contain a capsule. And remember, this is the sugary substance around the bacteria as part of the glycocalyx, which makes it difficult for phagocytes to engulf them. Now, host antibodies can counteract this, and this is why pneumococcal vaccines are effective in preventing pneumonias caused by Streptococcus pneumoniae, for example. So let's talk about some other innate host defensives, starting with inflammation and fever, which are very important in host response to infection. Inflammation is a non-specific reaction to noxious stimuli. The telltale signs of inflammation, the cardinal signs of inflammation are redness, swelling, pain, heat, and also altered function. Proteins called cytokines and chemokines, these are the molecules that can draw white blood cells to the site of inflammation. Effective inflammatory response isolates and limits tissue damage, destroying damaged cells and pathogens. Inflammation can result in considerable damage to healthy tissue, particularly in key organs such as lungs and brain tissue. So see, we have a localized infection, which leads to localized inflammation in this part of the body. followed by hopefully healing. What's fever then? Fever is initiated when certain cytokines, particularly the cytokine IL-1, interleukin-1, will cause the host's body temperature to rise, causing a fever. So when you have a fever and your temperature rises, it's due to release of this cytokine by your own white blood cells. Your own white blood cells release IL-1. This goes to the hypothalamus of the brain and resets your body's thermostat to a higher setting, a higher temperature setting. Why? Well, fever-causing cytokines, these are called pyrogens like IL-1, because they generate heat. They generate heat. A fever is beneficial. Your body wants to have a fever when there's an infection, because a fever is beneficial to you. It increases circulation, which allows those white blood cells to get to the site of infection. It's also beneficial because some pathogens cannot tolerate the increased temperature. And it's also beneficial because fever is associated with an increase in transferrins, which means these transferrins sequester iron. They keep iron away from the pathogens, limiting their growth. Remember, iron is a growth factor. And without readily having access to iron, microorganisms slow in their growth rate, which allows your immune system to catch up and clear the infection. So what happens during a systemic inflammation event or systemic septic shock, right? This is when you have more than just a localized infection, or more than just a localized problem. This is a widespread, that's what systemic means. Widespread systemic inflammation can lead to shock, as the increased vascular permeability decreases a host's blood pressure. This is called hypovolumic shock, right? You've got the decreased blood pressure or decreased fluids in the blood, and you've got shock, which can cause damage to multiple organs at the same time. Gram-negative bacteria are particularly dangerous because remember, they contain LPS in the outer membrane, which triggers a pro-inflammatory cytokine response from leukocytes. As their toll-like receptors are activated, this leads to a cytokine storm which can be fatal. So here you can see that in this case, the inflammation is not localized. It's a systemic infection leading to inflammation throughout the body and disease throughout the body and fever. Next, let's talk about the complement system. Complement are a series of serum proteins that play an important role in the immune system. So they're actually, complement or C prime is a set of circulating inactive proteins that can get activated in response to a pathogen. So think about it. These are not cells. These are a series of different proteins, serum proteins, that can become activated and help fight an infection. It's really neat how they work. What you need to take away from this slide is that there are three ways by which these complement proteins can be activated. There's what's known as the classical complement activation response, where basically in response to an antibody, if there's an antibody attached to an antigen surface, this would trigger activation of complement. So that's your traditional classical way that these complement proteins become activated. Next, there's what's known as the mannose-binding lectin complement activation. This is due to more of a nonspecific event where the mannose-binding lectin binds to the bacterium and that activates complement. And then there's this alternative complement activation as well, which is independent of antibodies. I don't need you to know exactly how these three activation events occur. Just know there are three mechanisms: classical, mannose-binding lectin, and alternative complement activation. These are the three ways by which complement becomes activated. And then once complement is activated, complement activation leads to three ways by which the bacteria are destroyed. One is called the membrane attack complex, which is direct attack, membrane attack complex, MAC, which we'll be talking about. One is recruitment of phagocytes with chemoattractants C5a and C3a. And one is through opsonization of the microorganism with C3b. So again, once the complement proteins are activated by one of three methods, right? Classical mannose-binding lectin or alternative, they can undergo three avenues by which they can destroy or help destroy the pathogen. And that is through formation of a membrane attack complex, recruitment of the phagocytes, remember neutrophils, macrophages, dendritic cells, or opsonization, right? These are the three ways. So we're going to talk about these three methods of attack, right? We're going to talk about them. So here you can see once complement becomes activated, one of three ways, it is the complement protein 3b. Complement protein 3b can attach to pathogen and trigger endocytosis, phagocytosis. So 3b is known as what's called, you need to know this term right here, opsonization. Opsonization is when a protein or molecule attaches to a pathogen, marking it for phagocytosis. You could even opsonize a toxin. It doesn't have to be a pathogen that gets opsonized. So when I say opsonized, I mean a molecule binds to something, marking it for the phagocytes to phagocytose, to engulf, right? So look what happens once complement gets activated. Complement protein C3b attaches to the surface of a pathogen, and that marks it for phagocytosis. And that process of marking something for phagocytosis is called opsonization. So I want you to know, for example, on an exam, which complement protein is known for phagocytosis or for opsonization. You would say that would be complement protein C3b. I hope that makes sense. Now, here's another avenue by which these complement proteins can destroy bacteria or mark bacteria. Here you have, again, you have activation one of three ways. And in this case, you have these different proteins called C5b. Complement protein 5b will insert itself into the membrane of the target cell. So this would be like a bacterial cell. So complement protein 5b inserts itself in the membrane, followed by complement protein 6, followed by complement protein 7, followed by complement protein 8, and a series of complement protein 9s. You see how many complement protein 9s are attached here? And once all of these are in, this is what's known as the MAC, or membrane attack complex. It's essentially, they formed a hole, a pore in the membrane. What happens when you form just a big old pore in the membrane of a cell? Well, that means that solutes from inside can diffuse out, solutes from outside can diffuse in, and the cell is going to get lysed. It's going to get destroyed from osmotic stress. Does that make sense? So the membrane attack complex, what do I want you to know? I want you to know that the membrane attack complex includes complement proteins C5b, C6, C7, C8, and a series of C9s, and this forms a pore called the membrane attack complex, which ends up lysing the cell. All right, what other ways are these complement proteins useful to the cell? Useful to, I should say, your immune system. Complement activation can lead to complement C3a and complement C5a, and these are attractants. These are chemoattractants. These will attract, what are they called? oh, let me see, C3A, C5A. Oh, C3A is more of a, well, C3A and C5A can degranulate mast cells. Let's talk about this one first. C5A and C3A can degranulate mast cells. Remember, mast cells are granulocytes which release histamine. Histamine is very important in inflammation. Histamine is very important in vasodilation. That means dilating of blood vessels. Okay, histamine release. So C3A, C5A, this is histamine release. Okay, and also chemotaxis. C3A can lead to chemotaxis as well. I just wanted to show you exactly. Let me see here. Yes, so look at here. Binds to the target. C3A, which diffuses into the surrounding area and serves as a chemoattractant. So C3A can serve as a chemoattractant. It attracts the neutrophils and other phagocytic cells to the site of the infection. Does that make sense? So again, what I want you to know, again, I want you to know C3b is complement protein for opsonization. C5b, C6, C7, C8, and a series of C9s is there. Once activated, forms the membrane attack complex, which lyses the bacterial cells. And it's C5a and C3a, which are involved in inflammation, due to histamine response, histamine release from mast cells, and for chemoattractant, right? To attract the neutrophils to the site of the infection. All right, here again, this is part of the the alternative pathways. You've got the mannose-binding lectin and the alternative pathway of activation. The mannose-binding lectin, or MBL, and the alternative pathways rely on innate mechanisms rather than antibodies like the classical activation method to activate the complement pathways. While they start at a different place in the pathway, all three complement systems end with opsonization, direct attack, remember that's your MAC attack, and recruitment of phagocytes and histamine response too as well from the mast cells. Some chemoattractants, C5a and C3a, are considered anaphylactoxins because they can lead to immune overactivation and potentially anaphylactic shock. So when you hear about anaphylactic shock, it's because of some of these chemoattractants like C5a, C3a, which can lead to immune overactivation in some people. All right, now let's talk about another component of the innate immune system. These are called the natural killer cells, NK cells, which are technically lymphocytes, but not part of the adaptive immune system, not part of specific immunity. That's chapter 27. These natural killer cells are still part of the innate defenses. So what happens with these natural killer cells? Natural killer cells are cytotoxic lymphocytes. Cytotoxic means they can kill your own cells. These are your own cells that are sick. Why would you want to kill your own cells? You'd want to kill your own cells that are sick. Natural killer cells recognize cells that do not display major histocompatibility complex I proteins, as all normal cells should have this protein. So this is a protein you need to know about. It's a receptor protein on the surface of your cell called major histocompatibility complex I, or MHC1. All of your nucleated cells in your body should have MHC1 on the surface. The only reason a cell would not have this protein on its surface is cells can internalize the MHC1 protein. That means bring it in from the surface when they realize that they're sick or something is wrong with them. Did you know that your cells have the ability to kind of sense when they're healthy and sense when they're infected or something is wrong? When the cell senses stress, when the cell senses it's been, let's say, taken over by a virus, the cell will internalize its MHC1 proteins, and natural killer cells can sense this. Natural killer cells are screening for this. If a cell lacks the MHC1 at the surface and displays a protein called the stress protein at the surface, which it normally should not show, that shows that the cell is in stress, a natural killer cell will activate to destroy that target, which is likely a virally infected or cancerous host cell. See, if a cell becomes virally infected, or if a cell is becoming cancerous, that could trigger internalization of the MHC1 proteins as well as expression and presentation of a stress protein. This is recognized by the natural killer cells, which will jump to action. So a natural killer cell will activate when it sees a lack of MHC1 and the presence of a stress protein. Activation leads to destruction of the target cell, which is likely a virally infected or cancerous host cell. How? By releasing granzymes, an enzyme that induces programmed cell death called apoptosis. Apoptosis means programmed cell death. So basically, the natural killer cell will tell your infected cell to kill itself. And also, the natural killer cell will release what's called perforin, which is a chemically and functionally similar protein to C9. It pokes holes in and perforates the target membrane. Remember C9, complement protein C9, which kind of forms a hole, C9. So perforin is kind of like the Mac C9, but it's produced by natural killer cells. So look here. Here is a normal, happy, healthy cell on the left and a diseased or distressed cell on the right. This is one that's either cancerous or infected by virus on the right. This is a normal cell on the left. Let's look on the left. Here, the normal, healthy, nucleated cell, like let's say your skin cell, I don't know, your skin cell, it presents MHC1, major histocompatibility complex I on the surface. When this MHC1 is on the surface, it's recognized by the MHC1 recognition receptor on natural killer cell, and there is no stress protein. The stress protein receptor is not, it does not sense a stress protein on the surface of the normal cell. So this normal cell checks out. This normal cell is spared its life. However, on the right, check it out, you've got a diseased target cell. This is a cell that has, like I said, been diseased in some way. In this case, it has internalized the MHC1. It does not have MHC1 on the surface. It is also expressing what's known as stress protein, which is recognized by the stress protein receptor. In this case, the natural killer cell knows there's something wrong. It will release its granzymes. It will release its perforin. Perforin pokes holes in the diseased cell. Granzymes promote apoptosis, program cell death, and this cell will kill itself before it can become a problem. Okay, and lastly, let's talk about another really interesting part of the innate immune system called interferon pathway. Interferons. So what are interferons? They are an innate response, an innate defense against viruses. So interferons are small cytokine proteins that are produced by virally infected cells, and these prevent adjacent healthy cells from being taken over by virus. Interferons serve as a warning system and prevent viral replication by stimulating the production of antiviral proteins in uninfected cells once they receive the interferon signal from the infected cell. So let's see how this works. It's very straightforward. Let me explain it to you. On the left, you have a virally infected cell. See, on the left, you have a cell that's been infected with a virus. That's not good. On the right, you have an adjacent healthy, happy cell that needs to be told that, be careful, there's virus around. So let's start our story on the left. Look what happens. A virus infects this cell, takes over the cell. Remember what viruses do? They start replicating inside of the host cell. This results in signal transduction, which allows the cell to recognize that it's been infiltrated. The viral component synthesized and assembled. See, the virus is assembling itself. The virus is assembling itself. However, the cell has kind of realized through this signal that it's infected. That's not good. So what does it do? It expresses interferon. It transcribes and translates interferon protein. So expression of interferon genes and the synthesis of interferon, IFN. Interferon is a chemokine. It's secreted from the cell and it travels to adjacent cells, you see? So secreted interferons diffuse from infected cell and bind to receptors on neighbor cells. So it's paracrine signaling. You have interferon now attached to interferon receptor on adjacent cells. Let's say this is an adjacent healthy skin cell. Now you have signal transduction. Signals go to this nucleus, telling the nucleus to start producing antiviral proteins. Proteins that will degrade viral RNA, that will, and also a lot of times transcription and translation of viral DNA can be blocked. You can destroy the viral RNA. Do you see what happens? So here's what I need you to know. Interferon does not prevent this cell, this second cell, from becoming infected with virus, but it does prevent the second cell from being taken over by the virus. Does that make sense? So that leads us to the end of this chapter. I hope this was interesting and exciting. The immune system is fascinating to me, and I hope it was also fascinating to you. Let me know in the comment box below if you have any questions, and as always, I'll see you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.